Thank you for the introduction and welcome to everyone, especially the members of the External Advisory Committee. I hope your symposium is productive today. And by way of introduction, I just wanted to take you on a brief tour of a disease that I see, because I'm going to make 3 points in the evolution of this disease. It's a disease, while important, was not even known until the 1950s. It took 30 years for the genetic basis of it to be identified. And then today we're still struggling because the genetics of it only explains part of the phenotype. And my point is that continuing to learn about these diseases has real clinical significance. We were actually chatting at breakfast about some of the new devices that we have to treat people, but we're still learning the best way to use them. Let me just tell you in 2 or 3 minutes the story of this disease, because it turns out this disease called hypertrophic cardiomyopathy, not an infectious disease, but hypertrophic cardiomyopathy is the most common inherited cardiac disorder. It occurs in about 1 in 500 people. If any of you are or were Boston Celtics fans, you may remember in the Larry Bird era, there was a player by the name of Reggie Lewis who actually died while shooting hoops from this disease. And hypertrophic cardiomyopathy turns out to be the most common cause of sudden death in non-competitive athletes. So it is something that is important, especially on sports teams, and yet many of you probably never heard of it. And in fact, the medical community hadn't heard of it until the 1950s, when a cohort of young people who had died suddenly were actually studied, and they found out that these people had abnormal thickening, hypertrophy of the heart muscle that led to fatal arrhythmias and sudden death. It took, so at first the disease was, oh, isn't this interesting? What causes it? And we had no idea for 3 decades until Cricket Seidman and her colleagues at Harvard identified a dominant mutation, a missense mutation, that was associated with this disease. And we've now learned that mutations affecting about 8 or 9 genes that code for sarcomeric proteins cause the disease. So that was the phase where we thought, oh, we've got this figured out, we now understand. Except let me fast forward to the present, where it turns out that there is a huge difference between the genetics of the disease, the genotype, and the phenotype. And we understand a lot less now as to why a patient who may have the gene, the genetic abnormality, has no morphologic evidence of the disease. So I actually saw a patient like that last week who has a mutation in one of the genes, and her heart, as near as I can tell, is normal. So what do? How do we treat her? We're not sure. My point in this introduction, in this tale of disease, is we went from a stage of not understanding to thinking that we understood everything, and we're now at the phase where it's complicated, and we need to try to tease the relationships out. So I say to you, welcome to this symposium as you try to figure out some of these important relationships. As we move knowledge, though, it can go from not understanding to thinking that we understand everything to a stage of, gee, it's complicated. It isn't quite what we thought when we thought we figured it out. And to finally get it right and understand it, we may go through phases of not understanding, understanding, but maybe not fully understanding, and then eventually figuring it out. So I urge you to have perseverance as you study your area of interest, and hopefully you'll reach that phase where we really do understand the mechanisms involved, so that then we can feed that information to people like me, so we can do something to help patients in the real world. So welcome to the symposium. I'm going to be in and out because of my other responsibilities, but I hope you enjoy it. Thanks again to the EAC, and I wish you a great conference. Thanks.
Yeah, if you guys want to put a little pool down here, I'll have it.
So you know at state fairs when they have people jump from 35 feet, 40 feet, 50 feet, you know 80 feet to 80 feet of water.
It's a good summer though.
Yeah.
Everybody wants to try it out. Yeah.
But today he's going to tell us some really cool stuff about T-cell immunology.
Good morning, everybody. I assume you can hear me fine. Can you hear me too fine? Okay, this is okay? All right. I was hesitating to put it here because then you get the in and out thing. I'm there. Thanks to the organizers. Thanks to the Cobrae for inviting us out. This has been good. That's a lot. Sorry. Looking forward to a good time talking to people today and tomorrow and hearing more about the program here. My specialty expertise is in really the boundary between innate and adaptive immunity. So the major goal of vaccine development is to essentially provide you with immune memory prior to ever having encountered an infectious agent at all. And we can do that a couple different ways. We can make vaccines from an actual infectious agent that we can attenuate, which earliest vaccines were. That's the way vaccinology started, way back when, before it ended up becoming the smallpox vaccine. And the other way to do it is to steal bits and pieces of the infection and put it into a subunit vaccine that we put some components into to try to alert the immune system it's time to pay attention. There were a lot of rules we got from the infectious process that we thought we could sort of adapt immediately into the vaccinology side of things. And what I'm going to tell you about today is some of the rules that we're learning that it turns out maybe that's not nearly as easy as we thought it was, because maybe there's a slightly different set of rules that the two are based on. So this is sort of the basic idea, is that we've got, can you see that? Let's see over there. Oh, there. An infectious generates a T-cell response, a CD4 and CD8 T-cell response. And I'm going to be speaking about vaccinology from a very T-cell centric perspective. So we make vaccines very good for mounting B-cell responses. And in fact, pretty much all the vaccines you've ever encountered in your life, the reason you're protected is because of neutralizing antibodies. That's why you have good vaccine-elicited protection. But there are other sorts of conditions in which we need T-cell responses. Essentially, any condition you already have, if you already have the infection, for the most part, making more antibodies isn't going to help you get rid of it. Thinking of Hep C or HIV or TB or malaria or cancer, in all of these conditions, these are more chronic style conditions. In those cases, we really need to generate a robust T-cell response. An infection does that extremely well, for the most part, assuming you survive. And the belief was that all we really need to do is take whatever the rules were here that mediate this. The more we study this infectious response, the better we can adapt those rules into the world of vaccinology. And one of the things that became clear about 10, 15 years ago is that there are a whole host of receptors that are pattern receptors that identify molecular differences between infections and between us. And those activated receptors actually mediate this transition between innate and adaptive immunity. And so now that we know what the number of those, the stimulators of those innate receptors are, if we take those and throw those into our vaccine of choice, we should be able to recapitulate whatever the necessary signals were that made this transition from innate to adaptive. We should be able to mediate it in this fashion without the actual infection. And this is what we call, in the vaccine world, the adjuvant. This is derived from Latin, "juvare," which means to help. It's what Charlie Janeway called the immunologist's dirty little secret many, many years ago. And it's the thing that basically induces the necessary immune activation that alerts the system, or we wish it to alert the entire system to pay very close attention to whatever antigen we put in and mount a very robust response against. So those adjuvants come in a variety of classes. Some of them are very defined. Some are very defined agonists for these various innate receptor families. There's a variety of them that we've used for many, many years, even though we're just starting to understand what they do and how they do it. There's a bunch of other ones that do a bunch of things we still don't really understand, and some various formulations that seem to be very helpful in eliciting different kinds of immunity. Now, the interesting thing about these is that if you take those into an animal model and ask the question, how well do they elicit T-cell responses? We know quite a bit about the B-cell responses, but how well do they make T-cell responses? And specifically, CD8 T-cell responses are ones that I, for the most part, study, although we study some more responses as well. If we take an artificial antigen, ovalbumin, which is the best antigen known to humankind for making CD8 T-cell responses in a mouse model. So if you can't make a response to ovalbumin, you can't make a response to anything, basically. And if chickens ever turn pathological on us, we've got it down. They know how to immunize against ovalbumin, so we're all going to be fine. Mix in your adjuvant of choice, shoot it into a mouse, and then using flow cytometry, ask the question, how many T cells did you make as a result of that vaccination? And the answer is actually painfully few. When you look at the antibody responses, this is just sort of an example where you can take antigen like anti-Figol and study the antibody response against that over a number of days. And if you pick TLR agonists of various kinds, you'll pretty much always improve that antibody response. So pulling things from the infectious agent and mixing it in as our adjuvants works very well for getting better and better B-cell responses. It turns out the same is not true for CD8 T-cell responses. And this is something we noticed, so I spent a few years in pharma at 3M, back when I did TLR agonist program. They developed a bunch of TLR7 agonists. And this was very surprising to us because if you looked at, so this is flow cytometry for those who aren't familiar with this. Without going into a lot of details, the number of dots in the box in the upper right are the number of T cells we made as a result of this vaccination. So the more dots, the better, basically. And this was surprising to have so few because if you looked at the amount of innate activation, we could tell that the adjuvant was doing something. It was inducing tons of inflammation. The inflammation seemed to be proceeding as we expected. But it wasn't transitioning effectively to an adaptive response. It really didn't matter which TL agonist we picked. We could pick a host of them. That didn't seem to matter. It didn't really seem to matter if we combined them. In fact, sometimes depending on how we combined them, things got worse. And none of them were as good as an infectious agent. At least 10 to 50-fold reduced responses as an infectious agent like vaccinia or LCMV or one of these other viral models could generate within the same 7-day period. So the infection was still doing something that our adjuvants were not. And we sort of ended up with a handful of principles. Even though these innate receptors induce a lot of inflammation, they produce incredibly small T-cell responses that doesn't seem to be commensurate with the amount of inflammation that they generate. And so it seems silly now, looking back on it, but at the time we began to develop the conclusion that actually inflammation doesn't equal an adjuvant. That is, there's a lot of inflammation that seems to be around for the sake of more inflammation. And then there's a certain kind of inflammation that somehow effectively transitions between the innate and the adaptive responses. And that's a very clear difference, and it's actually the one we need to camp out on for some time to figure out what are those signals that mediate that transition effectively, and what are the signals that are just a whole bunch of inflammation that doesn't really get us. So this is sort of, we spent quite some time sitting on this and publishing some papers on it. And as I said before, we really believe that if we watched the infectious response and understood the rules of the infection and all the factors that were important, we would gain insight into how to make our vaccines better. But interestingly, what we found was that there's a bunch of signals that the infectious response really needs to see. And then it turns out there's a bunch of signals that the vaccine-elicited response, the non-infectious vaccine-elicited response needs to see. And if you start removing them from the vaccine response, the entire thing comes crumbling to the ground. But the infectious response doesn't really care about those factors in the same way. And that was quite a bit of a surprise, again, at the time. In the last 5 years, we started maybe coming around with some ideas as potentially why that is. We don't know for sure, but we may be at least generating some reasonable testable models for it. So what I'll talk about today is a number of these factors that we found that the infectious response, it turns out, doesn't really care about at all. If you get rid of them, an infection will generate a T cell response perfectly fine. But if you get rid of that factor, the vaccine response comes tumbling to the ground. And what does that mean about how the infectious response, or the vaccine-elicited response is built? Because at the end of the day, the kind of vaccines we want to make are not really attenuated infectious agents, right? We want to make subunit-based vaccines. They're certainly much more controllable. There's a lot less toxicity. You can't attenuate everything. If you've noticed, but you can't attenuate HIV. It's unlikely we will ever be able to attenuate HIV or TB or malaria or a variety of things. So our future, we would say, lies in the direction of subunit molecular-based vaccines. And if the rules of those are incredibly different, we probably need to stop looking at infections to give us a whole lot of insights, and we need to look more at successful subunit vaccines and ask what the rules of those are. So I'm not going to go into a whole lot of details on the vaccine we used. We played around with this for quite some time and ended up coming with a combination that was kind of startlingly good. And in fact, using simply two adjuvants, two different adjuvants that target two different pathways, within 7 days we can recapitulate the magnitude of the infectious response just without an infection. So we target a molecule called CD40, which is on dendritic cells, it's on B cells. It's been known for quite some time that when you stimulate CD40 in a dendritic cell, you'll get some pretty robust immunity of a variety of kinds. And what we found a number of years ago now is that if you, it's a little hard to see here, if you use either a CD40 agonist or a TLR agonist alone, again, you get these very minimal responses. There's just not a lot there. But when you put the two together, you get a tremendous synergy. Okay, so targeting simply two molecules in the innate pathway allows us to recapitulate the magnitude of the infectious response without the infection. And this is true synergy in the sense that 1 + 1 = 30 here. It's definitely not additive. Interestingly, there's a handful of principles about this that are sort of highlighted here. We can pick any innate agonist, we can target any innate pathway or any innate receptor, and we get the same kind of synergy. So I'll summarize a handful of things here that we learned over the years about synergy, this synergistic interaction between these two adjuvants. One is, like I said, we can pick pretty much any innate stimuli, from toll-like receptors to nods, to ALPs, to a variety of cytokines. There's a variety of ways of innate pathways that will synergize with the CD40 pathway. This elicited robust CD8 and CD4 T cell responses, so we can get T cell immunity, augmented T cell immunity of both varieties of it. And this wasn't just something that we saw. There was a number of other groups that were also publishing on this and showing that this combination was a very, very successful way of making very, very strong CD8. So we had a system that was very reductionist. It made large T cell responses. So now we can ask the question, okay, what is this response built on? When we start removing factors from the host, when does our T cell response come down? And again, we had always taken cues from the literature on when people have been doing research on LCMV or vaccinia or listeria. What are the rules that those are based on, the T cell responses, that those infectious responses are built on? And we'll see if things are happening the same way here. And one of the first things we found that started kind of putting a hole in that wall was something called IL-27. So I won't go into the reasons why we picked this up in the first place, but IL-27 is a cytokine that is very pleiotropic, and when we use pleiotropic in immunology, it means it does a whole bunch of things that seem counterintuitive and we frankly don't quite understand. It's both pro-inflammatory and anti-inflammatory. It seems to depend on when it's present. Early in response, it stimulates a molecule inside the cell called SCAT1, which is very pro-inflammatory. And later in response, it stimulates SCAT3, which is very anti-inflammatory. In fact, in the world of infections, if you remove IL-27, mice tend to die. But they don't die because the infection becomes rampant. They die because the immune response. There's so much T cell expansion in these hosts, they die from lymphoproliferation. So the trouble, right out of the gate, IL-27 seemed like it was a factor whose job it was to pull in the reins of the T cell response in the back end and make sure it didn't go hog wild and kill the host. Not a factor which was important in the initial stages of getting off. But for a variety of reasons, we thought, well, we need to check this out. So we took a look at some IL-27 receptor knockout mice. I guess we wouldn't be standing here if we hadn't found some sort of phenotype. So what we found is that the response to our vaccine adjuvant was incredibly dependent on IL-27. And if you got rid of IL-27, the vaccine response took a nosedive, but the infectious response was completely unaffected. Okay? And this was true whether you look at percentages, the total numbers of T cell responses, the vaccinia response, or the listeria response, or the LCMV response, pick an infectious agent, you kind of found one that doesn't have, if anything, a larger T cell response in the 27 receptor knockout than the wild. So this was somewhat surprising, but very satisfying. We're now starting to see that we have a factor that we know is really centrally important in vaccine-elicited response. And that's interesting if our kind of freakish combination vaccine happens to work this way. But it's helpful if, like, most vaccines work this way. It turns out that it doesn't have to be the combination vaccine. It doesn't matter which adjuvant you pick. We can pick any number of a handful. We haven't yet found one that isn't IL-27 dependent. In vital cells, singly or in combination, all of the T cell responses drop pretty substantially when you get rid of IL-27. And this is, we used bone marrow primer experiments and Pathogen transfers to show that this was a T cell intrinsic phenomenon. Cytokines can do lots of things. They can not only affect T cells, but they can affect antigen-presenting cells. And so if you mess up the antigen-presenting cell, you will then mess up the T cell response. And it turns out that we knew that the IL-27 those signals had to be received by the T cell in order for them to become competent to expand. So I'm not going to go into a whole bunch of details on this. We published this a while ago. So I'm just going to summarize a handful of data showing that, just describing what we realized is that SCAT1 and 3 were both necessary. The T cell had to receive these signals. And if it did, over a period of 7 days, it would proliferate wildly and survive. But if it didn't have IL-27, what we found was that it wasn't that it didn't proliferate wildly. The proliferating wildly part was always intact. It was whether or not they survived that. So up through the first 3 or 4 days, the proliferation of cells that don't have IL-27 is perfectly normal. And it's between days 3 and 7 that they tank pretty hard, and most of them die off. It leaves you with a 10 to 20-fold deficit. So that's very interesting because that tells us something very unique about the process by which this is occurring. The cells aren't having a hard time dividing; they're just having a hard time living. And those are two very distinct problems that we can go after. So the questions then become: A, how are they dying? And B, how are they living? What's keeping them alive? I'm not going to go into details on this. We think necroptosis is involved, but I'll just talk a little bit here about the back end about what we think is keeping them alive. And so we took a look at the surface of the cells and asked. The T cells that do or don't have IL-27 receptor, do they look any different? Do they express markers that would be consistent with some sort of survival signal? So as I said before, if you look at the ratio, if we take a mouse and transfer in wild type and 27 receptor knockout T cells, and then immunize, and we can look at the ratio of those two to ask how things are going between the two. As I said before, at day 3, that ratio is 1 to 1. So 27 receptor knockout cells are doing just fine early on in the response. But by day 6 and 7, the ratio has already gone to 10. So somewhere between here, we have a massive drop in survival. And right around that inflection point, somewhere between days 3 and 5, we start seeing differences in a molecule called secretion. So the expression of this was very much lower on IL-27 receptor knockouts compared to wild types. So what's CD122? CD122 is a cytokine receptor that is part of both the IL-2 and IL-15 receptor system. Its job is to receive signals either from IL-2, which has an alpha, beta, and a gamma chain. This is the IL-2 receptor beta. Or it can receive IL-15 signals. And IL-15 is a little weird. and that it's a cytokine that's trans-presented. We think of cytokines, most pro-inflammatory cytokines, as being released into the external environment and floating around, eventually running to a receptor. But IL-15 is curious in that it's a trimeric receptor. It requires both the beta and the gamma chain of IL-2, but it also requires an IL-15 receptor alpha chain. But that has to be actually produced in the same cell that produces IL-15, usually an antigen-presenting cell. And when it does that, those are produced, exported to the cell surface, and actually trans-presented to the beta gamma on the T cell. So it's one of the only cytokines that's actually presented to the T cell inspection. So when you're looking for IL-15 production and activity, you actually have to look at the delivery cell. Once it receives this signal, there's a cascade of events that induce some transcription factors that are important to not only upregulating more of CD122 and maintaining its expression levels, but also promoting survival through some fairly reasonably well-classified things. So the fact that we were losing CD122 suggested maybe IL-15 was having an effect. Maybe what we were losing was IL-15 signaling to help keep the cells alive. Now again, this would be very counterintuitive from the world of infectious biology, because there's one thing that we've learned from infections like LCMV and Listeria and vaccinia, which is that T cells, during their primary response, don't care about IL-15. 15 receptor and 15 knockout mice were made 20 years ago, and they were challenged with everything till Tuesday, and all of them have a completely normal primary response. So what's affected is the memory response. As time goes on, memory cells are very dependent on IL-15 and tend to decline pretty rapidly, but the primary T cell response in an infection doesn't care about IL-15. So it didn't make a whole lot of sense to us why we would have a 15 effect that nobody else had seen one. But then again, we had a 27 effect that nobody else had seen. So the model was that maybe IL-27 stimulated initial amounts of CD122 that then could get reinforced by IL-15 stimulation. So we got IL-15 knockout mice and tried those, and again, lo and behold, IL-15 knockout looks very compromised relative to the wild type host, in pretty much the same way that the IL-27 receptor knockout looks compromised. And again, that's true not just for our combination agent, but that's true for all the agents we tested. And again, like IL-27, this is unlike an infectious condition, where there was a published paper from a number of years ago where they looked at 15 mice that overexpress 15 that have no 15 in the wild type. And in every single case, what's affected is the memory response late after the primary. But the peak of the primary response is completely uncaring about whether 15 is there or not. So again, we have another cytokine factor that the vaccine-elicited response really, really cares significantly about, whereas the infection is not. So I had my beautiful mind moment. You know, you sit in your office for like a week, and you end up with pictures all over and strings connecting it, and you don't bathe or shower, and your grad students get very worried about you. Just start suggesting counseling and medication. And this is what came out of that exercise. And I'm certainly not going to go into all of this. This is, if you look at the literature, at least if I look at the literature, this is what I came together with in terms of what's the transcription factor pathways that mediate a variety of aspects of T cell expansion and survival that eventually develop either into effector cells or memory cells. And what I'm going to emphasize here, you can see the red, these red portions were some of the things we started focusing on. One of which was IRF4 for a couple of reasons. IRF4 is a transcription factor that's really important in making good effector responses. It's been known in the infectious world that an IRF4 knockout looks actually, as it responds to an infection, looks just like our vaccine response in IL-27 receptor. An IRF4 knockout has a perfectly normal initial response. The cells proliferate just fine. They expand up to day 3 or so just fine. And then after that, they tank and die off. So there's a handful of other aspects that are very, very similar to our IL-27 receptor knockout response. So we decided to look and see is there a difference in whether or not our IL-27 receptor knockout cells have more or less IL-4. If they have less IL-4, maybe that's the pathway. Lo and behold, they actually do have less IL-4. And not only do they have less IL-4, but they have less IL-4 exactly at the time points that precede the nosedive that occurs in survival of cells right around between day 3 and day 6. And again, just to remind you, this is the sort of the inflection point from day 3. We've got equal numbers, and it's afterwards that we have a really significant change in the number of death processes. So at all these earlier time points, and actually if you go throughout, the IRF4 levels are substantially lower. So IRF4 looks like it could be central to this process, and we're in process right now to do some retroviral transductions and see if we can rescue this response by putting iron back in the cell. So if that's true, there's some other things that are downstream of IRF4. And again, interestingly, the things that are downstream of IRF4 are ones that the primary CD8 T cell response isn't supposed to care so much about. Two of them are T-bet and secretion. So these are two central transcription factors that are known to contribute to the fate decision of T cells. So some of you will be familiar with this idea of Th1 and Th2, a more viral type of response or a more parasitic type of response. And T-bet and eons are very important to tell T cells whether or not to move one direction or the other. They don't typically dictate the magnitude of the T cell response, but they'll sort of dictate the flavor of the response. But those are downstream of IRF4, so we ask the question fairly simply, what are T-bet and eons look like in our 27 receptor knockouts? And so again, we're taking wild type and 27 receptor knockout. These are called OT1 cells. They're T cells specific for hold-up, they're transgenic cells. Transfer them into a host, and at various time points afterwards, pull cells out and ask what's the differences between wild type and 27 receptor knockout cells. And we found substantial differences between T-bet and eons expression throughout these early phases. Substantially, this is a ratio of percent of max relative to wild type. Both of those are substantially reduced relative to wild type cells. So we have a significant reduction in these transcription factors that are downstream of IRF4. So it looks like those are all in line. So the next logical question is, well, does, in the same way the T cell response cares about IL-27, does it now also care about T-bet? Are these transcription factors also centrally important in managing the expansion of these T cells? And this is, I apologize, this somehow got reversed. These two plots are supposed to be like this. But you just look over here. There are mice that you can get that have both either knockout in T-bet or immunosuppression in the T cell compartment. And in both of those cases, we have a severe reduction in the response of the T cells. Again, similar to IL-27. Not quite as much. The defect isn't 10 to 20-fold. It's more like 5 to 7-fold. So that's sort of interesting. But we have a very significant reduction in the T cell response if you get rid of either of these two factors. And again, back to a theme. This is much along the lines of IL-15 and IL-27. If you look in the literature, when these were first published, if anything, sometimes the loss of these factors makes the T cell response actually go up, not down. But regardless, in the rest of the world, the response to an infection doesn't really care about whether T-bet eons is there during the primary response. Again, it will affect the flavor of cells coming out the other side, but it's not going to affect the magnitude. So now we have two more factors that the vaccine response really needs to have around the infectious response needs to care about. So that, for a long time, really bothered me. Like, that seemed really crazy. It didn't make a lot of sense. Certainly there doesn't seem like there should be evolutionarily selectable reasons why a vaccine response should be one way and an infectious response should be another. But as I got to thinking about it, we started thinking, actually, maybe we should have seen this coming, and maybe we should have thought of this a lot sooner and expected it. So when you stop and think about the course of an infection, an infectious response begins usually with the smallest number of infectious particles, virus or bacteria, and it grows substantially over about 5 days. Over that time period, the infectious process also grows. And there is a host of factors that rise and fall, and sometimes rise and then fall and rise again in response to this infection. And it has to do with how much the bacteria is dividing, the virus is dividing, how much inflammation it's producing, how many cells it's calling in, the various types of cells that are activated, and what pathways are activated by. But this inflammatory process, if anybody's ever opened up a host during the first week of infection, you'll know that it's a war zone. And all of these infectious processes, in some way or another, are contributing to the development of the T cell response. And at the very least, the T cell response is developing under these incredibly high inflammatory conditions. So if the T cell needs to see a STAT1 and a STAT3 and a TNF ligand and some sort of cytokine, well, it has probably a handful of ways of pulling it from the infectious milieu over integrating that into the curve of a 2 or 3 day period, at which to feed off of it and expand pretty dramatically. An infection, a vaccine, as we're talking about, is completely different. Its time course is dramatically different. We've done quite a bit of measuring on this. Much of this is speculative. The infectious posterior response, Laurel Lenz and the department helped me put together as best as we could put together. We've done some more careful measurement here. What we know is that a vaccine, unlike the infection, is peaking probably 4 to 6 hours after you initially inject it. And it's this synchronized hit of all the inflammation, all the antigen at once. And it peaks, like I said, the peak inflammatory response, as far as we can tell, is 4 to 6 hours in. And then everything dies off pretty rapidly in the greater scheme of things like that. To the point where within 3 days, most of your systemic inflammatory components are essentially gone. Now, locally, there's still some local inflammation that's going on. We have some reporter lists that we're starting to dig in exactly what those are. But the point is, there's a very narrow window of inflammation that's induced with the vaccine. And not only is it a narrow window, but it's a very specific set of infectious components. Whereas you may have a much broader activation of different innate pathways. Depending on which adjuvant you've chosen, you're going to have a much more narrow activation of innate pathways and inflammatory factors. And so maybe it stands to reason, we should have seen it, that not only would the adjuvant be dependent, since it has so many fewer things to pick from, and such a much more narrow, it has such a narrow window to grab the inflammatory signaling benefits of all of these factors, and a much more narrow set of factors to pick from. Maybe it makes some sense that it's dependent on things very different than the infection, and even more dependent upon it's building a very, very precarious tower, and if you pull any brick out of any point, the entire thing is going to crumble. And it's all very metaphorical and very nice. But essentially, that's the broad idea we're moving towards. And again, it does make quite a bit of sense, at least with respect to sort of the inflammation generator. And it's interesting to note that IRF4 is known, these sorts of factors that drive the survival of effector cells, they're known to feed off PI3 kinase activation, which is downstream of almost the vast majority of cytokines. The inflammatory components tend to induce PI3 kinase, and that tends to induce things like IRF4 that helps maintain the survival. So I think we're probably on to something here. But we've obviously got quite a bit more to test. So ultimately, there's a handful of conclusions about, we're starting to dig into, so why IL-27, what's so important about it. It turns out a bunch of things are downstream of IL-27 that T cell really cares about. If you don't have IL-27 receptor in the vaccine setting, you don't have IRF4 activation, therefore you don't have Tdap, therefore you don't have immunity. You don't have survival, maximal survival of your T cells through the immune response. We've recently started looking at metabolic activity driven by IL-15, which has been in the literature for some time. We're actually finding some really interesting things about that. And again, we're finding that the rules of metabolism in a vaccine-elicited response are actually a little bit different than the metabolic rules during an infectious response. Again, given the different inflammatory milieu, maybe we shouldn't be so surprised. It's reinforcing the problem. But again, the broader idea that has come out of this last 5 years is just that we've spent all this time looking at infections that haven't necessarily told us all the right things we need to be looking at to make a better vaccine-elicited response. And in order to really understand those, we've got to probably go back to studying just adjuvants and comparing and contrasting them to the infection rather than trying to have the infection tell us what to do. And that should guide, I would say, ought to guide some of our screening in the future, right? So if we now know that IL-27 and IL-15 are really important in a vaccine-elicited response, well then maybe as we screen our adjuvants, we should be looking for ones that induce 27 and 15. And in fact, we have a reporter for IL-27 production that we made a little bit ago. And if you stratify the adjuvants based on how well they induce that reporter, generally speaking, they trend towards being the best adjuvants induce the most IL-27. So it's possible you could stratify better and worse adjuvants simply based on what kind of IL-27. Maybe a little bit more on it. But regardless, these sorts of studies, I think, should be informing what we're looking for as we screen for better vaccine. Okay, so this is the lab. This is our Joshua tree. See, there's a handful of people. I say that, they usually look like us. That know what I'm talking about. If you don't know YouTube, Joshua Tree, legendary album. This is our photo at the top of one of the mountains in Colorado. The data that I talked about was mostly done by a really talented graduate student in the lab, Mr. student, Nathan from OHSU, and Alicia was at OKU. A great lab for people that just lost one graduate student who just got his PhD, which is always both a victory and a little bit sad and tragic, but so be it. Have been collaborating very much with this particular data with Chris Hunter, UPenn, and always with Bob. So anyway, that's it, and I'd be happy to take questions. Yes, so the question is, what cell types produce IL-27? So predominantly, it depends on what you stimulate. So if you infect, Chris has been doing T-Gandhi work. So if you use T-Gandhi to infect a host, inflammatory monocytes seem to be the primary producer of IL-27. If you use an adjuvant, there are the XCL1 positive dendritic cells. So the bad F3 sensitive XCL1 positive. So they're the dendritic cells that are known to be really good at picking up dead stuff and presenting it to T cells, which is what you would kind of want. Is love. Yes. Yeah, so that we just recently verified the, do they produce 27? Yes, they produce more 27. Yeah, so cytokines are curious this way. They tend to have some sort of negative feedback loops, right? So if you get rid of a receptor, there's no sink. So they just kind of build up. So those mice, if you use a 27 receptor knockout, those mice have substantially higher amounts of, or at least detectable amounts, higher amounts of 27. Yeah. Yes, that's a really good question. So there are various signals in the immune system that are analog, and then there are others that are digital. So we know that, for example, CD28 stimulus on a T cell, more and more CD28 stimulus you give it, the better the T cell response tends to do. But then there are other ones like CD27. That seems to be digital. You got to get enough across the bar. Once you do, if you add more and more and more CD27 stimulus, actually you may start killing stuff. So it's not clear at this point. We're sort of stratifying adjuvants based on IL-27, but we don't know where that must plateau out somewhere probably. We're not totally sure where the steep part of the curve ends and the flat part starts, and how analog versus digital is. That's a really question.
So, also curious in terms of your IL-15 and 27, they have a nice response. We move when there is no immune response, but we move even when you have vaccine response. But the T cells, you kind of change slightly when you have knockout versus
Yeah. So there's a handful of things that are different. Things like CD22 expression. IL-27 seems to be necessary to induce other things like LY6C. Weirdly, they have more CD25 or less CD25 expression, but they express more IL-2. Nobody's ever gotten rid of that one. So that's weird. So there's a handful of differences, and the question is which ones is it? My recollection is that there is a SNP, I don't remember which one, in WSX1, that the IL-27 receptor, there's a SNP there that segregates with something. But I honestly don't remember. I think Chris told me. I don't know that. But wouldn't that be cool? I mean, not cool. Yeah.
Yes, argument kind of induce limited for robust response. But just like LPS can induce tolerance, these arguments, if they induce tolerance, then it will not be not adequate response as compared to like infections. So, these arguments induce tolerance.
Yeah, so if it induces tolerance, it's not an adjuvant. So that's one way of looking at it. Adjuvants are, by definition, the things that avoid tolerance. So it's very clear in a lot of work that if you introduce an antigen with either no adjuvant or insufficient adjuvant, you induce tolerance, right? A T cell that encounters its antigen outside of the right kind of inflammatory stimulus eventually dies off, and all of its future dies with it. So are there better or worse adjuvants that induce more or less of that? Yes. There are opportunities to induce tolerance therapeutically, right? So a lot of immunity would be very helpful to do that. And there's a number of techniques that are being used right now, and interestingly, those are all techniques that are introducing high doses of antigen with as little adjuvant activity as possible. So I guess philosophically, I would say that if it's inducing tolerance, then it's not natural. Yeah.
Try to screen the other use. What do you find?
Well, so we found 27, we found 15 as a result of seeing what we did. We found 27 because we knew it was stat 1 dependent. So using some stat 1 with the 3 knockouts, we knew the T cells had to have stat 1. And trying to figure out what were the stat 1 related cytokines. We knew type 1 and type 2 interferon were T cell didn't care about those. They're great cells. And when you look through the literature, what's a defined stat 1 activator, alpha-7 pops up pretty quickly. It's like, we avoided it for a long time because the literature says, no, it's involved in immune suppression, in fact, it has nothing to do with immune activation. And then it does have something to do with pushing cells towards Th1 and away from Th17, that's that one stuff that most of the literature didn't seem to suggest this. So we eventually got it, but that's how we got it. Do they say it? Do they? In terms of the vaccine response? Or their cytokine? Right. Yeah. Right. Correct, yeah. So
I was asking whether it's common.
yeah, so CD122 knockouts have really high levels of 2 and 15 in their system. So there's a lot, so that's other ones like that. Gamma receptor knockouts have higher than that. Interferon receptor knockouts have higher than 12 because interferon and 12 are obviously good. So you play with a cytokine, usually another one goes out with the same. Interferon receptor knockouts have higher levels. Yeah. Yeah, there's not many of those published, but it's sort of like those everybody seems to know about. So we play around with OVA a lot because of the OT1. All the tools to analyze the response are easily available and they're very good. We've used multiple other antigens. We haven't found anything to be antigen related. If you have a good CD4 or CD8 epitope, you'll get it. Yeah.
Are there any labs looking at using environmental to prolong that activation period of the vaccine?
So, yeah, and some of the earliest techniques were, right? So you put it in an emulsion or something. You put it in a depot. And for many, many years, it was thought from the earliest times when Alexander Glenn was doing this with allergy. It was thought that depot is important, that slow leaking of antigen from that site is an important thing. For B cell responses, it's not so much important, but they don't care so much. It's fine if we do it. 2 years after Glenn published this stuff in 1925, he showed that you could cut out that site and have the B cell responses fine. The curious thing about T cells is that if you have a peripheral antigen depot, they like to go back there and they think it's an infectious site and they try and kill stuff. And in the process, they die vigorously. So all of the magnitude of the T cell response that I've made here, I can get rid of it completely by just putting it into a emulsion. So if I do that, it tanks the whole thing. So the formulation is actually really important. And a lot of those early ideas about, well, let's prolong the inflammation if we can, that tends to work poorly with regard to T cell. Now that's like, could you put like a non-antigen very depot over here and an antigen over here and have this inflammation? People played around with that, but the efficiency never seemed to play out. Yeah? Say again? So that one isn't. The 27, well, the GP130, yeah, the 27 is part of the IL-12, and GP130 is a shared portion of the receptor. So that's the stat 3. But the IL-27 itself is not shared with, one of them shared is with IL-35. So a lot of those shared receptors. All right, thank you.
Okay, so thank you for the invitation noticed a few things. I'm going to always expect the weather to be like this in November whenever I come. You have the most beautiful sunsets I've ever seen, for sure. And I've been really honored by the welcome because if we look here on the neuroscience building, I saw a truck name, a spider piece with underneath 1905, which was the day that, or the year that truck name and caliber was identified. So I've been very impressed that that was on your neuroscience building. So I'm going to talk about the path that we've had towards syphilis vaccine development, and it's been a very long path. So I'll give you a little journey here. In terms of the burden of this disease, most people don't talk about syphilis every day. I do, but most people don't. So I'll just give you a little rundown on the burden of disease. There's about 36 million cases with about 11 million new infections every year. This is mostly in developing countries, but not solely. In terms of the US, over the last 4 years, there's been about a 19% increase in the number of cases. And this is matched by countries, high-income countries around the world, Canada, Australia, quite a few countries in Europe as well. And if we look at North Dakota, zeroing in a little more, there's been a 437% increase over the last 4 years here. And that seems huge. And it actually is, but you've gone from a very low number of cases, 8 cases in 2011 to 43 cases last year. So if we look at Grand Forks Herald, just this year there have been 3 articles alone that have talked about syphilis. So STDs on the rise in the nation and the state, the 3 top ones, gonorrhea, chlamydia, and syphilis. In April, sexually transmitted diseases up again, syphilis and gonorrhea. And then this one, syphilis rates up among women in Minnesota. And that's actually fairly unusual. So usually in high-income countries, it's amongst men who have sex with men populations. So there's something different going on in Minnesota. The burden of congenital syphilis also is the most common infection associated with fetal loss or stillbirth in developing countries. There's about an estimated 1.4 million pregnant women infected per year. This is likely a real underestimate because a lot of these pregnancy losses are not reported or not understood what the cause was. And it leads to either stillbirth or loss, and also babies that are born with clinical syphilis, which is extremely debilitating, mentally debilitating, and physically debilitating. In the US, it's gone up 38% over the last 2 years. In China, we've seen an even bigger increase, up to 70 cases per 100,000 live births. And this is being mirrored around the world. So it is a public health concern. It's got a low infectious dose. It's about 50 organisms. The chancre that arises at the site of infection is painless. So in men who have sex with men and in females where it's internal, people often have it, they don't even know it. The disease enters the latent stage, so people are infected, and again, they don't know it. And then in terms of the interplay between syphilis and HIV, it increases the risk of acquisition and transmission of HIV 5 to 7-fold. And this is not only because you have a large chancre that arises, which allows for transmission of the virus, but it's also because there are biochemical characteristics that are upregulated within syphilis infected people that cause an increase in susceptibility to HIV. So I like to look at syphilis vaccine development in terms of the need, the design, and then the future, and we're going to go through that in the talk. So in terms of need, there have been several large-scale programs that have been implemented by WHO And so you should be able to get rid of it from the world, but in fact, it's not worked out that way. And so despite these intensive public health campaigns, where they're doing increased screening, increased treatment, the cases are still rising. The diagnosis in early infection and late infection, the tests that are currently available are very poor. It's impossible to differentiate between active versus previous infections, and the current diagnostic tests that are used in the US, they rely upon patient return compliance, and that often doesn't occur. The clinical complications with this disease, it's a wide range of symptoms that present, and so it's difficult for clinicians to always accurately diagnose this infection, especially recently where there's been a lot of cases of things such as ocular syphilis, people are going blind, and it's because of syphilis. And it's the highest value of all of the curable STIs. So if we can prevent this disease through vaccination, it will have the greatest impact amongst the curable STIs. And surprisingly, there's a global penicillin shortage. So even in the US, it's difficult to get your hands on penicillin in certain areas right now. And this is mirrored across the world. And that is the only truly effective way to treat this disease. And then economics, it's estimated that it's about $1 billion public health dollars in the US alone that are spent every year on syphilis-related disease conditions. So that's the need. There's a need for a vaccine. What we're doing currently is not working. In terms of the design, this is where it gets a little bit more difficult. So what we have done is looked at the progression of the disease that occurs in untreated patients. This is work that was done many years ago that stemmed out of unethical human trials, but there were these discoveries that were made. So we have infection and the disease enters the primary stage where you get chancre. This completely resolves. It's a large lesion, completely resolves, and then the disease enters the secondary phase, which is characterized by a rash. Then that also resolves, and the disease enters the latent phase. In the majority of individuals, the disease remains latent for their lifetime, but in about 30% of infected individuals, you get tertiary syphilis, which is characterized by several typical symptoms, including tissue destruction, which is called gamma. It's not a cancerous tissue destruction, paralysis, tabes dorsalis, and cardiovascular syphilis involvement. So what to take out of this is that the ability of the organism to disseminate very widely is large, and the organism actually invades every organ and tissue very early in infection. So early on, you get invasion of the central nervous system, crossing of the placenta, and then invasion into the bloodstream through the endothelial barrier. So if we're able to target this dissemination capacity of the organism, we have a chance to prevent transmission, to prevent congenital infection, neurosyphilis, and the disease symptoms that arise from this disease. So that's what we have been focusing upon for the past 20 years. My lab has been looking at this. And when you look at the organism that causes this disease, Treponema pallidum, subspecies pallidum, it's a spirochete. And it has very high invasive capacity, as I've described, but it has no known virulence factors. So it doesn't have any of the classic virulence factors, classic proteases, classic toxins. The genome was sequenced within the 1990s, and over 30% of the open read frames were unique to T. pallidum and did not share sequences with other proteins in databases. So if that still holds true today, it's an extracellular pathogen. And there's some other complications with this pathogen. It can't grow it in culture, makes it difficult to work with. As I said, there's no classic virulence factors. You can only recapitulate the symptoms in rabbits and humans. And rabbits are an outbred population which has its advantages, but also disadvantages. And the generation time is 33 hours, so you don't get many organisms out of the end. The organism is very fragile. It's got an unusual outer membrane, so it makes it difficult to work with. It just falls apart if you try to do any sort of conventional experimental techniques. And it's got a very low protein content, so it's impossible to identify what's in the outer membrane and interacting with the host using regular conventional methodologies. So where do we start? And we started quite a number of years ago looking at the parallels between invasive bacteria and cancer cells. And they called them metastatic bacteria. And there were a few examples in the literature of these bacteria, kind of at a high level and not looking at the molecular mechanism that was involved. But there was this similarity between cancer cells and binding to basement membranes, and specifically to laminar basement membranes. So that was where we started our journey. So for tumor cell dissemination, you often have binding to laminate basement membranes. You have through signaling mechanisms, you have opening of tight junctions. The cancer cells then invade into the bloodstream and spread from the site of the primary tumor. And we hypothesized that Kedl did a similar thing, and that we have binding to laminate, opening of tight junctions, intravasation into the bloodstream, dissemination, and then extravasation into the tissues. So the very first thing we did many, many years ago in the 1990s was to look at the complement of open reading frames, which is fairly small. It's about 1,000 open reading frames that are encoded by this pathogen, and try to predict through bioinformatics what was present on the surface and interacting with the host potentially. So there were just a handful that we were able to identify back then with those tools. And we expressed those in a recombinant form and then tested for binding to laminate. And found one protein, 751, that bound to a high degree of laminate. And when we looked at the expression of this protein on the surface of T. pallidum, we had to use indirect measures because you can't do conventional experimentation with this pathogen because it falls apart. So instead what we did was we looked indirectly at opsonic phagocytosis. And this is where we take macrophages, we take virulent treponemes, and we take sera against specific proteins. And then we test for the ability of the macrophage to bring in the bacteria. And so the concept here is that if the protein is on the surface, the antibody will bind, and then it will engage the Fc receptor on the macrophage, and it will be brought into the macrophage. So we can count the number of bacteria within the macrophage and use that as an indirect measure of surface exposure for a protein. So when we looked at positive controls, we had immune rabbit serum, we had sera against a known outer membrane protein, the only known outer membrane protein, BAM-A, which is found across gram-negative bacteria. Those showed high levels of opsonic phagocytosis, and 751 was even higher than that, suggesting it's on the surface, and these are our negative controls. So we also tested for binding to other host components that are found within the proximity of the vasculature. And this protein binds to fibronectin, fibrinogen, and then this is laminin again, so it's binding to proteins within the vicinity of the bloodstream. And what we also looked at was the ability to bind to umbilical vein endothelial cells as well as microvascular endothelial cells. And we just did a plate-based assay where we're growing these human cell lines in plates, and then we're coming on top with histidine tagged recombinant protein, And then we're testing through nickel HRP for the binding. And we're looking for the absorbance readings. And so when we did this is showing 3 different constructs of 751, different portions of the protein that we expressed. And they all bound to high levels compared to a negative control protein, which was minimal. And this is showing endothelial cells and microvascular endothelial cells. So we always want to take it back to the organism. We can't do that with T-Palatin because you can't grow it and you can't do genetics. So what we did instead was to express this protein in Borrelia-related spirochete. So we took a non-infectious Borrelia that is fluorescing, expressing GFP, and we did this in conjunction, in collaboration with the Moriarty Lab at the University of Toronto. So what they did is to express 751 on the surface of Borrelia, and this was in conjunction with John Leong's lab at the Tufts University. They showed through FACS analysis that the protein was expressed on the surface of Borrelia. And what we then did was took those surface expressing 751 Borrelia and looked for the ability of those bacteria to attach to endothelial cells through static assays and then through shear flow assays where it's mimicking the bloodstream. So in static assays, this work was conducted by one of my graduate students, Karen Lithgow, and she looked at the ability of these Borrelia to bind to endothelial cells using a plate-based assay again, and just looked for fluorescence and found that compared to the parent Borrelia strain, we got a higher amount of the 751 expressing Borrelia attaching to these endothelial cells. And then we've also looked at this in an in vitro bloodstream model. This is work conducted in the Moriarty Lab in Toronto. And what we see is an increase in 2 different constructs that are expressing 751 on the surface of Borrelia compared to the parent strain, and it's approximating the level that infectious Borrelia bind to the vasculature, and also Borrelia expressing a known vascular adhesive, UBK32. So I'm just going to hopefully start a little video to show you something else. Maybe not. So we also looked at this in vivo within a mouse. And what we found was a similar phenomenon that the Borrelia that are expressing 751 bound to the vasculature of the mouse in vivo. We looked at this through intravital microscopy and saw that in comparison to the parent strain, there was a huge increase in terms of vascular attachment by these Borrelia. And in the video that I was going to show you, you can actually see the bacteria adhering to the vasculature and the flow of blood. It's really interesting. If anybody's interested, I can show you after. So recently we've identified the structure of this molecule, and it belongs to the lipopelin superfamily, which is interesting because eukaryotic lipopelins play a role in dissemination of cancer cells. And they do this through signaling pathways where there are decreased expression of E-coherin, the junctional protein, and opening of cell junctions, and then invasion of tumor cells into the bloodstream. So we were pretty excited when we saw this structure. And what we wanted to do at this point was to use proteomics to try to identify what the host receptor was that this protein, this trafficking protein, is binding to in the vasculature. So what we did was a pull-down assay where we took 751 and mobilized it onto a resin. And then we had endothelial cell membrane proteins that we isolated. from both brain microvascular endothelial cells and vein endothelial cells. And we pass this protein preparation over the immobilized column and then use LC and SMS to try to identify what bound to the column, what was present in the alveolus. And we found something interesting, that we were consistently pulling out the laminin receptor. And we've done this 3 times, twice with vein endothelial cells and once with the brain microvascular cells. We're getting the same result being pulled out. So the laminin receptor is interesting because it interacts with integrins, and it's involved in signaling pathways that result in cytoskeletal rearrangement and opening of tight junctions. And it's been found to be the receptor for several highly invasive pathogens, including Neisseria meningitis, Haemophilus influenza, and strep pneumonia. And so Karen again has looked at what happens to an endothelial layer when 751 is exposed to that endothelial cell layer. And what she finds is that it's disrupted. The endothelial layer is disrupted. This is in comparison to a layer that is exposed to a controlled recombinant protein. So if we come back to the structure, we've been able to map all of the host binding capacity of this protein to one face of the protein. So it's all along here, both the host cell binding capacity as well as the ECM binding capacity. And then T. pallidum has this unique alpha helix that kind of caps the structure. And that is unique to TP0751. We think it may have a regulatory role, we're not sure. Because when that alpha helix is present, there's no effect on host cells. And when we take that off, we do get the effect of the opening of the tight junctions. We also get secretion from host cells, which I didn't have time to go through. And the other thing that we notice is during infection, this protein actually, it's longer than this. It's got a long end-terminal tail. And it's actually degraded down to just this lipohealing core. So we think there's processing that's going on during infection, and that this is the functional domain that causes the effect on host cells. So lastly, I'm going to talk about what we have done in terms of testing this in an animal model. So we have taken groups of rabbits and we've immunized with this protein. We've then challenged with T. pallidum, and we've had a control group of rabbits which have not undergone immunization. And then at day 15 post-challenge, we euthanize these animals, and we look for the treponema burden within various tissues and organs within the rabbit. We also take lymph nodes from those animals, and we introduce those into naive animals, and we look for the development of infection within those naive animals. And we test this via the development of orchitis in the testes, because that's where they are introduced, and seroconversion. So the concept here is that if the organism is not able to disseminate because you have immunized with 751, then you will not get a productive infection in these naive animals. So in terms of qPCR, when we look at the treponemal burden in the organs and tissues, I'm showing 3 tissues here, bone, liver, and spleen. We've analyzed other tissues as well and see the same effect. In the immunized rabbits, which are shown in blue, you have a decreased treponemal burden compared to the control rabbits, which are shown in red. And overall, you're having a decreased burden within these immunized animals. And then when we look at the transfer experiment, in the animals that receive the immunization, you're having an increased cellular infiltration in those animals compared to the control animals. And when you look at the transfer rabbits, 0 All of them were uninfected, as shown by apparatus development and seroconversion. So seroimmunity was generated in those animals. And both of the control animals became infected. And this cirrhosis early occurred about 17 days after transfer of the lymph nodes, and we took these rabbits out to 6 months with no evidence of infection. So what we think is occurring is that through 751 immunization, we're able to cause an increased amount of 751 specific antibodies to be present. And then we're having increased opsonophagocytosis, which is the primary mechanism for clearance of this organism during infection. And then we're also blocking this attachment to the vasculature. And that's preventing the organism from gaining access to the bloodstream and to disseminating and then seeding into these external sites of infection. So interestingly, when you look at the structure of 751, it's matching the structure, as I indicated, of several proteins, lipocalins, eukaryotic lipocalins that are involved in tumor cell invasion. And it's also matching the structure of the factor H binding protein from Neisseria meningitidis. And this is a protein, the only protein that is shared between the two commercially available Neisseria vaccines, Trumemba and Vaccinology. So we think this might be a bigger phenomenon in that this lipocalin domain could be acting as a scaffold to cause this protection to arise for invasive pathogens as well as tumor cells. So in terms of this being a vaccine candidate, we've shown it's on the surface of the pathogen. We've shown that it binds these extracellular matrix components that are close to the bloodstream. It's a vascular adhesion. We've shown that in vivo. It has this structure that induces a signaling cascade. You see a drastic effect, visual effect on host cells. And through immunization, we're able to inhibit this dissemination. So that brings me back to the future, which now that we have this candidate vaccinogen, we need to move on to try to entice agencies to support this and to consider it as a vaccine. So this has been surprisingly difficult. So what we need in this is an advocate from regulatory agencies and from industry to take this on. There's still a lot of stigma associated with this disease, and we have to overcome that. We need dedicated funding. These are very expensive. It's an expensive disease model to study. And we need new investigators. Right now there's 5 people that study this pathogen on basic science level in the world. And there's a few of them that are about to retire pretty soon. So we need an influx of new people to come into the field. So just to acknowledge the people that have really done this work, the people within my lab, also my structural colleague at the University of Victoria, and then the Moriarty lab at the University of Toronto, and then my funding NIH. And this is showing an image of my lab when we went to play disc golf on a mountain. And we ended up finding this wooden bar that somebody had built on top of the mountain. So we were there the second year having a drink up the top of this mountain. So thank you for your time. There's a lot of
which is really unique to this protein, or?
Yeah, so the question is, there's a lot of redundancy in spirochetes in terms of attachment to the host. So the genome is much smaller in T. pallidum compared to Borrelia. It's about a third the size. And so it's a real evolutionary genome reduction, example of that. And so everything that it has, it uses efficiently. And there are other adhesins for sure that we know contribute. We don't know anything about the expression during the different stages of infection. And so we're wondering if there's some sort of regulated expression at different stages. We know from our detection results that it seems this protein seems to do something significant. And maybe the other adhesins are expressed later, we just don't know at this point.
Did you try to do RNA-seq to kind of like monitor the expression this way?
So RNA-seq on the trapnine?
Just to make sure, just to monitor.
Yeah, so the question is, have we tried to do RNA-seq on the pathogen to try to monitor expression of the protein? And we haven't. The problem is that we get so few organisms out, we can't get much material out of the rabbit. So we haven't done that. There have been some investigators that have looked at that, but it's always just a snapshot. So that's the problem. You're taking one snapshot in infection, and I think we're looking at it in a really unidirectional way, and it's actually more dynamic than we think. Because the pathogen can hang out for 50 years without, showing itself. So there's something quite complex going on here.
Question. So two questions, actually. So I was really intrigued with the apparent loss of ECAD here in endothelial junctions. Was that due to an actual physical interaction between the bug and the junction proteins, or is it due to a downregulation or loss of expression?
So let me clarify that. That is not something we have shown. That is something that is reported in the literature for invasive tumor cells. So we're going to start looking at that. We have not looked at that for our bacteria inducing that effect yet.
The second question was going to move to the brain barrier. Let's skip that. So what do you know what causes the latency to the reticular state?
No, so that is unknown, and it only happens in about a third of infected individuals. So it's got to be something to do with the immune system in those individuals that is causing some sort of re-engagement of the infection. And we don't even know, are the organisms different during that point? Whether the organism is doing when they're latent, do they look different? There's a lot of unknowns with this disease and this pathogen.
So, given how invasive these things are through extracellular matrix, just in tissue in general, I'm wondering if you think that without this protein, would the bug eventually be able to get over where it wants to go, even if it couldn't get into and out of the actual bloodstream at the same rate? I don't know what the term was. I think it was day 14 or 15.
So, if I understand the question correctly, if it didn't have this protein, if the pathogen didn't have this protein, could it still do what it needed to do?
Right, I'm trying to kind of separate the immunological protection from the inhibition of dissemination of phenotypes that the vaccine would have.
Well, so this protein is unique to T. pallidum, but we know that other spirochetes are invasive. They're not as invasive, in my opinion. And so there's motility associated with all these spirochetes, and that's why it's got the name and burrows in. So it's very adept at that, but I think T-column has taken it a step further with this protein to be able to facilitate this extra invasion capacity.
And do infected animals or people want to immune response against this protein?
They do, but it's low, and that is the characteristic of T-column, is that we think whatever it's showing on its surface, which is about 100-fold less than what E. coli shows on its surface, whatever it's showing, it has some sort of unusual capacity to not evoke a large immune response. We have an inkling through proteomic studies that it's due to post-translational modifications. We're just starting to look at that.
So, Carol, the protein binds laminin, and you think laminin receptor. Is there a laminin between the two? So is it coating itself with laminin and then triggering the laminin receptor that way, and so doing like cloaking itself and triggering its entry?
Okay, so that is a great question, and that is something we don't know the answer yet. But
Did it come up on the mass spec? Did laminin pop up?
no, it didn't. It didn't. So, yeah. So the other thing that we're thinking is in our bloodstream model, artificial bloodstream model, we start taking things out, and there is apparently, I've been told, there's a laminin knockout mutant of some of these cell lines, so we can take them out, test them in that, and then add things back in, and that's something we're going to start working on.
Anybody else? All right, our next speaker is Dr. Bill Kedl from the University of Kansas, and he's going to be talking about one of our favorite topics here among the microbiologists, type 3 secretion. And he assures me that he does not have any fun facts, that he's just a normal guy, hunting, fishing, those kinds of things. He did say that he has a monitor in hypnotism, so he'll try really hard not to put you to sleep. But he did come up with another potential fun fact, which is he's probably the only faculty member here who has won prizes at county fairs for the produce of his garden and products thereof, such as pickles or fermented foods, which is a distinct challenge to me. Now the gauntlet's been thrown, and you know me and know the giant set-block garden. So I'm going to have to try to best him now. Oh, the biggest zucchini is easy.
At the King County Fair. You will hear me okay? Right. I don't hear the microphone working, so I'm assuming it is. Okay. So actually, in the last 3 years after moving from Stillwater, Oklahoma to Warren, Kansas, and this is why I microbiology, because I actually chassis. I had to downsize from the garden. It was about 30 feet by about 7 foot down to the garden. It's about 20 feet. So I'm learning to cram things together. And I'm learning about microbial diseases that occur when you cram too many plants close together. So this is going to be a little bit different than the previous talks. We do have a goal in controlling disease, particularly since we're now in the Department of Pharmaceutical Chemistry and School of Pharmacy, where we have two main veins of research. One is small molecule research, with a lot of anti-cancer drug development, that sort of thing, a lot of formulation there. But also there's a lot of vaccine formulation stabilization goes on in our
department. And with the arrival of Wendy, my wife and I, to the department, that vaccine mission has expanded just a bit to include a little bit of stuff that's a little more early stage, antigen identification and target identification and so forth. My specific research is for the most part very basic. I look at structure-function relationships in secretion systems and how they work. Now, I don't think probably you can see or get on the slide all that well. Simply a number of different bacterial secretion systems. A couple that we focus on a little bit include the second one, the type 3 secretion system, as well as, to a lesser extent, type 4 secretion systems. We look at those in part because these are secretion systems that don't simply secrete toxins into the environment or enzymes into the environment. These are secretion systems that actually can allow a prokaryotic potential pathogen, not always pathogenic necessarily, but a prokaryote to specifically interact with and modify the behavior of eukaryotic cells. So our version of host-pathogen interaction is actually from the pathogen point of view and how the type 3 system, infected proteins can be injected into the target cell to modify its original behavior. For pathogenesis in the systems I'll talk about, mainly Shigalla, but also for other aspects of bacterial eukaryote interactions such as in rhizobia, the formation of ketones. So, the type 3 secretion system comes in more than 2 flavors, but 2 main flavors, and these are the flagellar type 3 secretion system. So basically, flagella and gram-negative bacteria are created because of a specialized secretion system that allows proteins to go from the cytoplasm to the extracellular part of the cell, where in this case they form a very long flagella filament, which allows for motility. We look at what's referred to as the virulence type 3 secretion system. And this is actually, you can see these here. These are purified type 3 secretion apparatuses. I think in this case they came from Shigalla or Salmonella. But these are specifically for modifying host cell behavior. They're not for motility. So I'm going to talk a little bit about Shigalla. All right. Now, one of the difficulties we have with Shigalla is the fact that this is a human-specific pathogen. There really is no good animal model. Higher primates can be used. However, if you look at the dose of Shigalla it takes to make a human sick, 1 to 200 organisms, compared to what it takes to make a rhesus macaque sick, which is usually on the order of 10 to the 4, 10 to the 5 organisms. So even the other higher primates, other than humans, are not the best model for human Shigalla infection. So in reality, Shigalla are simply pathobars known as E. coli, which is a huge group, as you all know. This organism causes shigellosis, which is bacillary dysentery. So it's not simply a watery diarrhea like you see with cholera and other toxigenic E. coli, but an actual invasion of the intestinal mucosa with resulting inflammation, ulceration, and symptoms of dysentery, which include initially diarrhea, eventually fever, scanning stools containing a lot of dysentery. Unlike most of the E. coli, this organism is not a motile organism, which is, from a model standpoint, is good, simply because we don't have 2 competing secretion systems. We have a single type 3 secretion system. No flagella system. Another thing that is important for prevention, and we hope for preventing shigellosis, is the fact that Shigalla have one absolutely essential gradients factor, and that is the type 3 secretion system. There are toxins that are produced, but in the absence of the type 3 secretion system, those toxins really don't come into play. Okay? By toxins, I include things such as set and sin, which are mild enterotoxins, but also Shiga toxins, which can be important during infection in children with Shigalla dysentery. So to look at this on a global scale, a recent study, the Global Multi-Center Study, the GEM study, was conducted by a group spearheaded out of the University of Maryland, the Center for Vaccine Development. And they set up 7 different sites in sub-Saharan Africa and South Asia. They monitored children, large numbers of children, for the presence of different diarrheal pathogens in school scenarios. Then they looked at that incidence and compared that to the incidence of diarrhea and dysentery, moderate to severe diarrhea, in those children. And 4 pathogens, not surprisingly for some of these, that showed up time and again were rotavirus, for which there currently is a vaccine, but also Shigalla, surprisingly Cryptosporidium, and then the enterotoxigenic E. coli, in particular stable toxin producing enterotoxigenic E. coli. And one of the big goals now of the Gates Foundation, as well as the path group in DC, is actually creating vaccines now that can combat at the same time Shigella and E. But in this GEM study, what they discovered was in a tiered system where they look at the incidence of these four different viral pathogens in the first year of life, second year of life, and then years 3 through 5, that not surprisingly, a major problem early in life is rotavirus. Okay? Shigella is in the top 4. In the second year, Shigella rises, still just behind rotavirus, but in years 3 through 5, Shigella becomes a major pathogen for children in these developing parts of the world. Now, this is not just to point out how Shigella is such an important pathogen, but also to point out the fact that look at all the players in the diarrhea cause in these developing parts of the world. Okay? So while there are on the order of, and the numbers vary depending on where you go, 100,000 and 500,000 deaths, okay, by shigellosis each year. Okay? When you consider the total picture of diarrheal pathogens, the impact is far beyond infant mortality. The outcome of that is stunted growth because of multiple episodes of diarrhea, okay? Cognitive disorders caused by disruption of normal flora, as well as repeated episodes of diarrhea. And so you see a major economic impact, not just because of illness, but also because infrastructure is so taxed trying to take care of these compromised children growing up, who will have debilitating effects throughout their lives. So how does Shigella cause disease? So typically this organism is ingested in contaminated water, which obviously makes rehydration difficult because the water used to rehydrate, as we call it all the time, is the same water that caused the disease to begin with. Okay? The organism passes through the stomach because it's highly acid resistant, uses these mild enterotoxins to quickly pass through the small intestine, and then actually sets up shop in the descending colon, the large intestine. Where the bacteria encounter M cells, the M cells are normally trying to sample the contents of the lumen of the gut, present that context continually to the immune response, the mucosal immune response here, the GI tract, and this helps maintain a healthy immune system overall. In the case of Shigella, however, the bacteria are quickly taken up by, or they enter macrophages, and they induce those macrophages instead of the macrophage winning the battle, the Shigella win the battle, and they induce pyroptosis. Okay? They also produce a parallel apoptosis, so you have 2 competing cell death pathways that are going on here. The result of that is twofold. One, the release of interleukin-1 beta, a powerful pro-inflammatory cytokine, which is ultimately going to contribute to some of the symptoms you see in shigellosis, but also allows for the release of the bacteria themselves. Okay? So while Shigella enters macrophages and induces the death of those macrophages, it is going to, once released, induce a similar type of response in epithelial cells. Okay? So it's causing these epithelial cells to behave like phagocytic cells, take the bacteria out. The bacteria quickly escape the vacuole, okay, that's formed by that uptake. Once they do, they actually become motile. Okay? They divide these epithelial cells, and they become motile by recruiting host cell actin at one end of the pathogen, and that actin then pushes these bacteria around these cells until eventually they're able to enter into a neighboring cell, lyse the resulting double membrane bound vacuole, and they set up shop and do the same thing all over again. So they move from cell to cell without ever leaving the safety of that intracellular environment. Meanwhile, you have all of these pro-inflammatory events occurring, and as these organisms begin to divide, you begin to have destruction caused by the inflammatory response, releasing organisms into the lumen, and they are then rich in that scanning stools that appear later in the infection process. So the type 3 secretion system is responsible for the majority of the events I just described. It's responsible for proper entry into macrophages. It's responsible for pyroptosis and apoptosis, responsible for entry into epithelial cells, is responsible for vascular escape. So ultimately, you get rid of that type 2 secretion system, you basically prevent this organism from causing disease. Now, my laboratory is interested in a couple of different aspects of the type 2 secretion system. The first part that I'll describe occurs at the tip of this needle, which is the extracellular portion of the type 2 secretion apparatus. We're interested in the presence of this protein called invasion factor antigen D at the tip of that needle that our lab initially described as being there. Also, the recruitment of subsequent translocator proteins to that tip where they directly interact with those cells. Then I'll say a little bit more about this structure that's at the base inside the pathogen side of the apparatus. So here's simply a comparison between the flagellar and the virulence type 2 secretion system. These types of apparatuses are present in a number of different organisms. Shigalla plexi, obviously, other Shigalla species. Salmonella has 2 type 3 secretion systems responsible for different aspects of the infection process. Yersinia, whether it be pestis or enterocolyticus or pseudotuberculosis, have a type 3 secretion system. Pseudomonas aeruginosa has one similar to the Yersinia system. Purpuraria pseudomallii, actually was part of our conversation yesterday about melioidosis, actually has 3 type 3 secretion systems. One is related to plant systems and 2 different types of animal type 3 systems. One related to the Yersinia system and one related to the Shigalla salmonella system. Okay? The first proteins recruited once there's post-cell contact are these hydrophobic translocator proteins. I'll describe one of these in some detail here. Once post-cell contact has occurred, these translocators form a pore in the target cell membrane, and it's through that pore then they can have secreted effector proteins enter the target cell to alter that target cell's behavior. So this work goes back probably a little more than 10 years, where we initially identified on the surface of the bacteria, in addition to this needle, it's comprised of a small protein called MCH. Some of you are familiar with proteins related to MCH like this guy. We discovered that at the tip of this needle, we could actually immunostain for invasion plasmid antigen D. Here's a case where we submitted the paper, they didn't believe it, D was at the tip, so they rejected the paper, we submitted it to another journal. In between time, Pete Kedl showed that LCRB was at the tip of the Yersinia. So, first we didn't do any papers there. We've got a step further in the Shigalla system, however, and this may not show up really well, but I'll do a little clearer in the next slide. We were able to show that not only is there a D at the tip, but that in a quiescent type 3 system, a newly made type 3 secretion apparatus, D is the only protein at the tip of that needle. However, if we incubate the bacteria with bile salts, we showed that we can induce the recruitment of the first translocator protein, invasion plasmid antigen D, to the tip of that needle, and presumably this then occurs to allow detection of post-cell contact later in the infection process. And the presence of it being at the tip of that needle requires the needle itself, the MCH protein. It also requires the D protein, that initial tip protein that we found at the surface. We were able to show that either needles that are associated with cells or isolated needles sheared through bacteria, we can do immunoglobulin labeling and show that both the and the B are present at the tip of the same needles. And most likely you have the D serving as a transition from the needle to the recruitment of the B at the actual tip of that needle. This was simply a set of experiments that we did using some fluorescent methods, where we actually put a fluorescent probe onto the oxycolate bile salt that we use in the lab, and we're able to show that the bile salt event, the thing that it binds to promote this recruitment, is actual direct binding to invasion plasmid antigen D, and we're simply looking at a change in polarization, that's rotational diffusion of the IPA-D, the fluorescent IPA-D, as we add unlabeled protein, and what we see is that there's an increase in polarization, which means a decrease in rotational diffusion, meaning that the molecule has become bigger and is physically bound now by a protein. That led us to a model we developed in fact, where we show that in quiescent needles, prior to the addition of bile salts, we know we have D present at the tip of these mixed needles, the Shigalla T3 secretion apparatus, but that's the only protein you can detect on the surface of those bacteria. If we add biosalts, we now can detect not only APD, but also APB at the surface, and these needles have not been induced for secretion yet. They are not secreting, but they now have this first largely hydrophobic translocator protein present at the tip of those needles. So we wanted to study APB a little bit further. APB is not an easy protein to study. We were able to purify and crystallize APD early on and have some models for how it may sit at the surface of the needle. APB has been a little bit more difficult to look at. Thanks to collaborators, Mike Barra, who now actually is in our laboratory, and Brian Kreisberg, who's then at the University of Missouri, Kansas City, is now at Kansas State University. They weren't able to isolate an internal fragment of APB, which they were able to crystallize, get a structure for, and basically found that it formed a very large coiled coil. Not a particularly remarkable structure, except that when you consider the proteins that are involved in the extracellular components of the type 2 secretion apparatus, the needle protein MIXH forms a coiled coil. APD, the first tip protein, is stabilized by a large coiled coil. APB, the next protein to be recruited into that complex, the internal third of that protein, the fourth of that protein, is dominated by this coiled coil structure, which led Mike to actually hypothesize that the APB N-terminus coiled coil may very well be involved in anchoring the APB to that tip complex, not so unlike how colicins are anchored via a membrane binding region, and then an active region which is responsible for the killing of other bacterial cells. And these are simply colicins from E. coli. So we think we have a model at least for how it could be made sitting at the tip of that needle, but we want to go a little bit further with that. Don't worry so much about this. This is simply some data that shows that prior to its secretion, APB is held by a chaperone, which keeps it from actually being toxic to the Shigalla organisms. And we were simply able to show that this internal coiled coil is required for and involved in that chaperone binding process. The chaperone will come in just a moment. Now, we wanted to know for sure, is APB, this internal coiled coil, responsible for anchoring this protein with APB at the tip of the type 2 secretion apparatus? So we did an experiment where we put a fluorescent probe onto this internal fragment of APB. So we have a fluorescent APB fragment. We incubated that with increasing amounts of non-fluorescent APB. And as we add the protein, we're looking at fluorescence polarization. Okay, in other words, a decrease in rotational diffusion as if the molecule is actually increasing in size. And what we see is, as we add unlabeled APD, we see zero change in the fluorescence polarization of the internal fragment. Unless we do the exact same experiment in the presence of biosalts, which we know bind to APD. In the presence of biosalts, we see a very nice APD concentration-dependent increase in the polarization, indicating an interaction between the two. Okay? And these are simply some different fragments, and what this simply shows you is that maximal interaction requires regions from 11 all the way out to about amino acid 200 in this process. Right? Now, in sort of a different experiment, we did another fluorescence technique called fluorescence resonance energy transfer, where we put two fluorescent probes on APD. One of these was at a tetracysteine group that we put within what we call the distal domain of APD. The other one was at a single cysteine present near the C-terminus of APD. We incubated this lovely fluorescent with a donor and acceptor probe to allow for energy transfer of APD with the internal fragment of APD, and saw as we increased the amount of APD, no change in the energy transfer between those two particular sites. However, if we include biosalts, you'll see in that process, we see a loss of energy transfer, meaning the excitation energy from the donor probe is not being passed to the acceptor probe. Okay, so we have a decrease in that energy transfer process, meaning those two regions are becoming further apart, meaning that the association between the two is causing a conformational change in the IPA. And what we believe is responsible for that is the fact that in some low resolution imaging that was done by Chelsea Pepper in our lab at Old Boston, they predicted that in the native pentamer at the tip of the needle, this distal domain appears to be crystal structure is being down, probably is actually flipped up. And these data would suggest that because these two probes are further apart as the rebinding occurs, it's this region that has that distal domain flipped up that allows for the interaction between the two. Okay? So we think we have a model for how the D is sitting out here when we have it at the needle tip and when the B is recruited to the needle tip. But we wanted to do some more stuff with full-length IPA. Now the difficulty working with full-length IPA, as I mentioned, is the fact that it's hypophobic. But we can purify it in large amounts as long as we purify it in the presence of a chaperone called IPGC. But if we want to look at the purified protein, we need to get rid of the chaperone. And we found that we can get rid of the chaperone by incubating this complex with detergents. We tried a number of different detergents and settled on a couple of very mild detergents. One is called LDAO, here the chemical name, oral dimethylamine oxide is LDAO. The other one is opioid octyl polyoxyethylene. What we see is that if we separate out that chaperone, get rid of the chaperone in the presence of the opioid, and then we cross-link what's left, we see that in the presence of opioid, the IPA forms a large complex. Okay? The precise size we did not know at that point. In the presence of LDAO, it shows up as a monomer. Okay, so the detergent can impact how that IPA translocator protein behaves. When we did analytical ultracentrifugation, what we found was that in LDAO, as we expected, it forms a monomer of the native molecular weight of about 65 kilodaltons. In the presence of the opioid, we see two species, one of which, the dominant one, is the tetramer, but we also see an octamer, which we believe is probably a dimer of tetramers. So we see two different potential forms of IPA protein here. Now, I mentioned these detergents were mild. These detergents are mild enough that you will not lyse liposomes, unlike something like Triton, which blows liposomes apart. Because these detergents will not lyse liposomes, we were able to look at the interaction of this IPA protein as a monomer or as a tetramer with liposomes containing fluorophores. A high concentration of fluorophores tend to be lynched for fluorescence. If you release the fluorophore from these liposomes, you see an increase in the fluorescence because you get rid of the iodine. What we found was that the monomeric form of IPA had no effect on these liposomes. The fluorophore stayed trapped. The tetramer, on the other hand, allowed for a very nice time-dependent release of fluorophore. So at this point, we thought, okay, the monomer will not interact with membranes, the tetramer will interact with membranes. Don't worry about this, but over here shows that we weren't quite exactly right. It turns out that if we do a flotation, okay, if we take protein, put it on a sucrose gradient, centrifuge that, we can push that protein to the bottom of the sucrose gradient. If we have liposomes, the liposomes float to the top of the sucrose gradient. If the proteins bind to the liposomes, they will float to the top of that sucrose gradient. And what we found was the IPA, which is no interaction membrane and opioid, would always float to the top of that sucrose gradient. Okay? On the other hand, where we expected LDAO not to interact with membranes, we found that the IPA and LDAO also went to the top of those gradients. So they're both interacting with liposomes, but only the IPA and the tetrameric forms allow the release of fluorophores. And the others are simply a number of different controls. For example, the IPA IPGC complex will not associate with liposomes. It goes directly to both the chaperone and the IPA go to the bottom. And these are some other controls over here. So we modified our fluorophore release assay to look at fluorophores containing fluorescent versions of different sized dextrans. And what we found was small dextrans in the presence of this tetrameric IPA were released quite readily from liposomes. However, as we increased that size of those dextrans, the more poorly they were released from the liposomes, suggesting that this complex, this tetramer, has pore-like properties. And what we're proposing this is actually the what I'm calling the pre-translocation pore, which is the initial pore inserted into the host cell membrane prior to actual reduction of effector receptors. So we have a model in which it typically is in a monomeric form. It can associate peripherally with liposomes. This may have something to do with inside the cell where IPA-B actually has an effector function. IPA-B will actually activate caspase 1. That's what results in pyroptosis we saw with macrophage. However, at the needle tip, it's a tetramer of IPA-B anchored to IPA-D via that internal coil that actually inserts into the membrane, form a pore, and this pore is through which then effector protein will be injected into the target cell. The size of this pore we estimate to be on the order of about 3.5 or so nanometers, which is consistent with the size, the internal diameter of that needle, which is about 2.5 to 3 nanometers. Okay, that's the whole thought. Not quite. Here's some newer data. So what we want to do now is we really would like to get some structural data on that whole group, if at all possible. So one of the things that we've done is we've identified potential root regions of IPA-B, regions that may be surface exposed, and we're actually inserting a phage T4 lysozyme insert into these sites. T4 lysozyme is nice because T4 lysozyme loves to crystallize. Okay? It is very nice for nucleating crystals for potential structural analysis. The other nice thing about T4 lysozyme is if you look at where the C and the N termini are located, they are so close to each other that if placed into a loop, you would expect minimal disruption of the overall structure of that particular protein. And in fact, I will do all of these, but I will point out that we have 12 different inserts now, the majority of which are actually active if you put those back in the Shigalla. So despite the fact we have this T4 lysozyme stuck into the IPA-B, the majority of the IPA-Bs are still functional, and for those that are not functional, we actually have reasonable evidence to show that they're not functional for very specific reasons, such as being at the N-terminus, they don't allow secretion, or being located within the N-terminal domain, they actually prevent chaperone binding, so the protein is proliferated. So this is sort of a direction that we're going with the IPA-B to try to get additional functional information or structural information to relate that with function. We're also using a number of different antibody-based strategies, such as using camel or PHH antibodies to try to bind to IPA-B, so we can also render it more soluble, so we can try to get some structural information. And in fact, we already have a larger fragment now that we have in crystal form that we're hoping to get some additional structure beyond that coil region. This does not include the transmembrane regions, which is one of the regions that we're very, very interested in. Now, this is what we're looking at the tip of that complex. So we have some low-resolution images of the needle itself with the D at the tip. We have some low-resolution images of this tetramer of IPA-B. This is done by cryo-electron microscopy. Very low resolution at this point. We're hoping to have some better images in the next 6 months. We can model the two together. Based on some quenching experiments, we feel that we have the N-terminal region anchoring IPA-B to IPA-D. The hydrophobic region is exposed. It's the hydrophobic region which has cholesterol binding activity that allows it to insert into host cell membranes, which often occurs at lipid rafts, which is distribution cholesterol and sphingolipids. And we think that is the initial interaction between the bacterium and the host cell. So, part of the reason we're interested in this complex is understanding how we can affect this complex to basically block its interaction with cell membranes. The other reason we're interested is for vaccine development. And that's work that's going on in my wife's laboratory, where she's actually looking at IPA-B and IPA-B as potential vaccine candidates, which is why I was very happy to hear Ross this morning talk about how the future of vaccines is really going to have to involve us in supplement vaccines, because that's the direction we're going. It's not a direction a lot of the rest of the things will be fruitful, but we should have broad slate now for phase 1 human trials, so it seems to be moving forward. So the overall activation of the secretion process, we think it is a 3-step process in Shigalla, where the lumen of the gut, the tip, When we have interaction with lipids, we now have to see the second translocator recruited to the tip. And at this point, we have a paper showing that indeed is the case. Once you have C recruited, the whole system is turned on. Okay? It becomes very, very messy to look at once you have the entire secretion process turned on. But we're not just interested in what's occurring at the tip, we're also interested in what's occurring at the base of the structure. Now I have a couple of videos, I don't know if you're going to work or not, we'll find out quickly. One of the things that we want to do is to be able to visualize this entire apparatus. So we're working now with June Liu, who was able to actually show flagellar structure in Borrelia, in part using spiroelectron tomography, because Borrelia is so thin. These spirochetes are very, very thin organisms. So he can get some very high resolution images of the flagellar apparatus in Borrelia. And so the goal here was to okay. So what we've done here is that we have introduced mutations in Shigalla that cause them to form mini cells. These mini cells are on the order of about 200 nanometers across. So now we're getting into a distance that's more similar to what you would see with the thickness of Borrelia. So what we've been able to show is from these mini cells, we can visualize multiple type 3 secretion apparatuses in these mini cells. And using cryo-electron tomography, can get higher resolution images of some of these things. Now we know that when we make these mini cells, that they are active, because we can take these mini cells, incubate them with red blood cells, and not only do we get the apparatus coming in contact with red blood cells, but we actually can insert pores that will cause these red blood cells to lyse. And so what we'll see here is here's a single apparatus which actually makes contact with this red blood cell. We allow that interaction to occur. We know that it will be or the translocation pore has been inserted, so we can disrupt that interaction, and now those red blood cells are lying. Contact hemolysis. Okay, so what we're doing now is we're using what we know about mini cell technology in June's lab to look at parts of the type 2 secretion apparatus that have not previously been visualized. So what June was able to show was that you can see the entire apparatus. You see the needle, which is on the outside of the outer membrane of the bacteria. We're also beginning to look at some of these same things with the tick complex, but I was discussing that here. But more importantly, in the cytoplasm, we are now visualizing structures that were not previously seen. Okay, previous structures and reconstructions have been done using osmotically lysed cells, okay, or purified apparatuses. And the majority of the material from the cytoplasmic portion of the apparatus are lost during these processes. Okay? What June was able to see, however, was not only do we see this transporter core in the center of the apparatus, but we're able to see that there is an extension down from the cytoplasmic membrane down to where the SPA47 ATPase that drives the secretion process is situated. And that ATPase seems to be associated with this six-fold symmetry type bulb structure. Okay, here are the pods of the bulb that you see there. And they seem to be associated with the ATPase via this bespoke life structure. And so now what we're wanting to do is dissect some of the events occurring down here. And this is not so much from a vaccine standpoint, but maybe from the pharmaceutical chemistry department, the possibility of identifying small molecules that might interact with and interfere with some of these protein interactions. Now, using a variety of mutants that we have, we use, for example, a mixing N mutant. And a mixing N mutant basically is one that is devoid of type 2 secretion. You have apparatus basal body assembly, but you have no assembly of any of the external forms. So there's no needles. Right? In these mixing N mutants, we continue to see, although it's more faint, continue to see these pods with a six-fold symmetry, but we are missing the internal parts. We're missing the ATPase. Okay, we then looked at another mutant, SPA43 mutant, and now we seem to be missing everything that's located in the cytoplasm of those particular cells. And so what we're trying to do is figure out what pieces sit where and how they sit relative to each other. So this is what we've come up with. We've come up with a model where we have this secretin pore top associated with the cytoplasmic membrane. We have coming down from the cytoplasmic membrane these pods, which we believe are composed of SPA33. They're held together then via these spokes, which is composed of links again, to situate the SPA27 ATPase and the associated stock in such a way that now you have recruitment of effector proteins, removal of chaperones, followed by secretion of the substrates, the protein substrates. So almost done here. In comparison with the flagellar system, I think we see some very important differences in the structural features, and we believe those are related to function. Where in the flagellar system, you have this C ring composed of fly-in and some other proteins. And here's a continuous ring. That continuous ring is needed for the rotational action of the flagellar apparatus, and eventually the rotational movement of the filament. In the case of Shigalla and other type 3 systems, we see this in salmonella now too. We believe we don't have this continuous ring, but actually the six-fold symmetry and these pods that are connected mechanically to the ATPase, which allows for a ratcheting and perhaps a movement of some of these parts, but no rotation of the parts of the base or the needle itself. And so now what we want to do is we want to look at some of the protein dynamics occurring part of the apparatus to figure out functionally what is going on, mechanistically what is happening. Okay? One more slide after this one that I'll end with. But first I want to acknowledge some really important people here. Some of this work was done in Oklahoma State University, especially in New York, and Phil Adams now with the Kansas Department of Health and Environment was important in some of the APP studies and interactions with membranes. Nick Dickinson is now at Utah State. He was very important in doing some of the HIPAA assembly studies, and he is now actually at Utah State, he's looking at structural features of the ATP associated with the base. Important players at the University of Kansas from the Department of Pharmaceutical Chemistry, as well as Brian Gaspar at Kansas State University, Jimmy Liu in Houston, and then Esther Lord was doing the low resolution images analysis at Boston University. So I'll end with a final video, which I won't talk about the video itself. It's sort of self-explanatory. It's everything that I've just described. This tells you everything that we know about the base of the structure. And now what we're wanting to do is actually be able to find probes that will bind to some of these components in the base. Because what I believe is happening is that there is an exchange between base components and cytoplasmic components that allow for a ratcheting of that via the ATPase of that basal structure, and that's probably what is driving the actual secretion process. So I probably didn't go over close. I'll be happy to take any questions that you might have.
Great talk, and one of the best structure functions studies.
The people on the far slide, they're the ones that they did all.
It's great. So my question is, IPA-B, is it, pardon my ignorance, is it immunodominant in humans? Yes. It is. Neutralizing antibodies?
Pardon?
Neutralizing antibodies?
Not completely neutralizing. Now, in the vaccine side of all this, it would be a very major part, and it would be as much more immunodominant as it is. But they both contribute because they do things together, they don't do them separately. And I would have to refer to our immunologist to more detail about that. The antibodies we generate are partially neutralizing the assays. I look at contact mediated hemolysis, but I look at invasion of cultured cells, but we don't see a complete neutralization. I do think during the natural infection that it's more than neutralizing antibodies in play. I think the CD8 response is important, the T-cell response side, I think is extremely important in this whole process. In current live attenuated vaccines, it appears that antibodies are dominant in protection, but those antibodies are always driven against the LPS. Okay? You will see an immune response against the proteins, but you really see it dominated by an antibody response against the LPS. And the problem with the LPS is the fact that we're dealing with 54 different subunit vaccine, because if we can make that immune response, recognize the proteins in a protective manner, we can cover all 54 serotypes. These proteins are so well conserved amongst all those serotypes. That's the perfect reason why we want to go a protein group right there, at the live table.
Much. This is extremely fascinating. I have a question that I'm interested in. So, do you see any evidence of some of these structures or molecules migrating into more adipose tissue? And have you seen any evidence of that? And when they go there, do you see any modification in the function?
Shigalla's a bit different. Any cell type that we look at in cell culture, they will evade.
Yeah.
They'll evade it around, they'll evade it.
But do they change the virulence?
Pardon?
Do they change in virulence or properties?
That we haven't looked at. But during natural infection, it's a very self-limiting disease.
Yes.
It never goes beyond this site.
Okay.
I don't know about adipose tissue. I do know that they can invade and or kill B cells. They will, in some cases, I believe they will also invade T cells in some cases, but it's all within these M cell, pirate patch like complexes. It's in the large intestine, most pirate patches are lower in the small intestine. There's still through these M cells that do all of this. There's so much going on and so many cell types involved that until the PMNs come in and basically destroy everything, they just kind of have their way with all the cell types.
Amazing. Thank you so much. Very interesting.
I just asked. So I'm curious in terms of the host response, if we normally remove the host cells, you still see the same amount of needles or there will be less needles down there?
Say that again?
So if you remove the host cells, in that case, you will see the less amount of needles formation?
The needles are all formed prior to actual contact and infection of those cells. So you don't really see, it's typically going to be between 50 and 100 needles per bacterium. I think the numbers are a lot less than you're seeing. Yeah, that's what I thought. So it depends on the system that we're looking at. In this system, whether there are host cells or not present really does not have a bearing on the number of needles that are present on certain bacteria. It can have an effect on the secretion status of those needles. Now, that's a little different from the salmonella system where there are two types of secretion systems. The one produced by pathogenicity item 1 are much like what you see in the Shigalla system, and they are made basically prior to host cell invasion. Once they invade host cells, though, those are turned off. Genetically, those are turned off. They turn on a second pathogenicity item 2. That apparatus comes on, and now that apparatus is all about maintaining that salmonella continuing back inside that target cell, whether it be in a epithelial cell or it's going to be disseminating bacteria, it's going to be a macro.
I was actually really intrigued by the structure. What I was thinking more in this regard was that will the bacteria take the ADP from the host cells to actually activate that thing, movement on that thing?
Now remember, this is the bacterial cytoplasm. And there is little, I would go as far as to say there's no real evidence that anything ever travels retrograde through that medium. That's basically proteins going in one direction. Is there host ATP contributing? Possibly. I think it's probably likely. Although one of the mechanisms that these bacteria use in the dissemination process, moving from cell to cell, is they will induce the host cell to secrete ATP. Neighboring cells recognize that, and that contributes to that cell-to-cell spread process.
Great talk. I was wondering whether there's a puppy, there are so many, right? So if you want to do vaccine, you want to do one of them will be most important. others who don't need it?
Okay, I think that remains to be seen, but there are 4 proteins that fall into this little class that was originally identified in the 80s before they ever knew about the apparatus that fall into the invasion pathogen antigen category. And HIPAA created HIPAA A, B, C, and D. It turns out that they were all named that because one, they're all expressed from a virulence pathway. But 2, during natural infection, you have a strong antibody response against all 4 of those proteins. So they were identified as invasion pathogen antigens. In our case, we know HIPAA D and HIPAA B are surface exposed. Okay? And that's why we have targeted those for potential vaccine development. HIPAA A, on the other hand, is a secreted effector protein. So HIPAA A never appears on the surface of the bacterium, although it can be secreted into the extracellular milieu. But it typically is injected directly into the target cell, okay, where it affects the active immunization process in the uptake. HIPAA C is sort of an interesting one because it's hydrophobic, it's part of the translocator, but I don't personally believe it ever appears on the surface of the bacteria. I believe it's injected into the cell where it interacts with the membrane, it's active and initiates the membrane webbing that results in pathogen uptake. Now, we did some work at Oaks and Walter Reed quite some time ago, where he was able to show that C very likely has an adjuvant effect on cells. And we've never really followed that up. Right now, we're just concerned with B and D. I am wondering if at some point, including the C in that process might be useful, not necessarily as an important antigen, but perhaps in skewing an immune response in a direction that might be useful in enhancing the immunogenicity. HIPAA B is clearly more immunogenic than HIPAA D, but when it next to HIPAA B helps HIPAA D become more immunogenic. And the cytokine profiles we see of them separately or together actually can be very different. So when there's the whole cytokine thing is so complicated.